Hello, good afternoon everybody, I hope you are well. Welcome back to Understanding Cancer Season 2. Today we're going to be doing Lecture 8 titled Types of Signaling Pathway, Normal and Dysregulated, PI3K, AKT, mTOR. So let's have a recap of what you should have understood from last week's lesson. So the EGF ligand binds with the EGFR receptor. Now this leads to dimerization of the receptor and autophosphorylation of the cytoplasmic domains. There are 7 effector proteins activated in signal transduction: GRB2, SOS, RAS, RAF1. This begins a protein kinase cascade with MEK followed by ERK1/2 followed by MAPK. Now, overexpression of these genes increased gene amplification and mutated proteins of the ligand or whether receptor or whether the adapter proteins or the effector proteins are implicated in various cancers. This can be range from prostate to breast to lung cancer to blood cancer to a variety of means. Now, crosstalk between GPCR and EGFR targets is via the members of the RAS family of proteins, RAP1 and 2, which are targets of pKa. One of the CAMP targets, EPAC, also stimulates RAP1 protein. So what will we learn today? So at the start we will discuss the structure of PI3K, which stands for phosphodilysyl 3 kinases. This is then followed by the structure of AKT, mTOR, types of mTORs, how is mTOR regulated, and we're going to go through the similar concept where we will discuss the receptor activation of this particular signaling pathway, how the signal is then transduced within the cell, and the types of cellular response. We will then discuss the link between EGFR and PI3K, AKT, mTOR signaling pathways. And the last part of this lecture episode will consist of discussing the causes of dysregulated PI3K, AKT, mTOR signaling pathway in cancer. So a gentle reminder, you can divide this lecture over the 7 days. It'll approximately take 1 hour, and you can then challenge yourself with a quiz. And how do we cap on how you can support your learning? We have diagrams by HN Designs presented in a simplified way. There is a glossary to help you understand any keywords. There's also revision posters. There's a quiz and a reference for further reading. So for acknowledgements, I'd like to thank my parents, the rest of my family, friends, colleagues, and the respective teachers and health professions who've basically installed the passion of cancer within me. So let's begin with the structure of PI3K. The PI3K, there are 8 types that are found within mammals, and they consist of dimers with 2 subunits. And these types of subunits are not the same, hence why they're heterodimers. The first subunit is catalytic P110, and the second subunit is called regulatory P85. Now these subunits can be divided into subdomains. So the P110 is divided into ABD, which stands for adapter binding domain, the RAS binding domain, RBD, C2, helical, catalytic kinase CAT. The P85 domain can be subdivided into N-terminal, SH3, row gap, NSH2, ISH2, and CSH2. So there's different forms of the SH2 there. This is a diagram by HN Designs, just to simplify matters and present to you where the subdomains are located. The C part is reference to the carboxylic acid position, because it's a type of protein, so it will have an amine group, which is the N-terminal, and it would have the carboxylic acid group, which is the C-terminal. And you can see both the C and the N-terminal can be found in both subunits, P85, P10 alpha. Now the PI3K has 8 family members, and it can be divided into different classes and based on the structure, regulation, and function. Class 1 particularly is divided into 4. So you have the PI3K alpha, PI3K beta, PI3K gamma, and PI3K delta. And there's class 2 and class 3. The PI3K class 1, in addition, can be subdivided based on the method of activation. So the PI3K is not a receptor as such. So the receptor tyrosine kinase, or the GPRCR, so G protein coupled receptor, are the 2 main receptors that need to be activated in order for the PI3K to be phosphorylated. Now, if the initial receptor activation begins with receptor tyrosine kinases, then the PI3 class 1 can be subdivided into IA, which means that it consists of the PI3K alpha, beta, and the delta. Now, if the receptor activation is by GPCR, then the subclass would be IB, and that would be PI3K gamma. So the PI3K class 1 can be divided into A and B, and then the subdivisions which we discussed in the previous can be entered within these 2 categories. So now let us proceed with the structure of AKT. So AKT is also known as protein kinase B, or PKB, and it consists of 3 family members, AKT1, AKT2, and AKT3. So all of these are type of serine threonine kinases, and they differ in the location where it is found. AKT1 is found within the cytoplasm, so where the chemical reaction takes place. It's also found within the nucleus, so this is what controls the cell, and both of these are apparent in a broad range of cell types. AKT2 is found in the mitochondria of many cell types, and this is necessary for apoptosis, so cell death, and also glucose metabolism for energy. Now AKT3, this can be found within the nucleus and the membrane that surrounds the nucleus, particularly in the brain, heart, and kidneys. So here you can see that the 3 structures of AKT1, 2, and 3 are presented, and each of these have a pH domain, which stands for the N-terminal plextrin homology domain. You also have the kinase domain, and that is where the catalytic activity takes place, so it's a central area, so it's the CAT domain. And you have the hydrophobic tail. Now the hydrophobic tail is essentially that it dislikes water, and it's from the C-terminal side, and it's also known as C-terminal extension, and it's like a regulatory motif. Okay. Now, if you look much more closely, you can see that the position where phosphorylation takes place within the kinase domain and the hydrophobic tail, it varies. So, for example, AKT1, the phosphorylation site is threonine 308, and the hydrophobic tail is serine 473. So, if you've noticed, these are the two main amino acids where are involved here. This is why they're known as serine threonine kinases, because they're a type of enzyme that speeds up the reaction, particularly at those sites, which is threonine and serine. Now, AKT2, the position varies, so rather than 308, it's 309, and rather than 473, it's 474. And for AKT3, the position is now 305 for threonine and 472 for serine. Now, despite this, there is a similar, or what we would say a homology, between the different isoforms of AKT. So AKT2 have 81% amino acids homologate to AKT1. AKT3 has 83% amino acid homology to AKT1. And there is also an additional position which is known as the linker region, and this occurs between the pH domain and the kinase domain, i.e. the pH and the CAT. And this area doesn't really have any form of homology to other protein kinases. So let's now proceed on to the structure of mTOR. So mTOR is an abbreviation for mammalian target of rapamycin, and it's an important AKT target. It's also known as FRAP, so FKBP12 rapamycin associated protein, RAFT1 rapamycin and FKB12 target, RAPT1 rapamycin target 1, and SEP is sirolimus effector protein. This is also a type of serine threonine kinase with a molecular weight of 289 kilodaltons, and it belongs to the family called PI3K related protein kinases. And there is a huge similarity between the C-terminus of mTOR and the catalytic domain of PI3K. There are also two types of mTOR, 1 and 2. So let's discuss the types of mTORs. So the mTOR C1, which is mammalian target of rapamycin complex 1, otherwise known as mechanistic target of rapamycin complex 1. It consists of mTOR, so you can see the mTOR where it's located there. It also has the raptor. So the raptor, that is what regulates the mTOR protein activity, and how it's done is by a process called phosphorylation, where there's an addition of a phosphate group, and phosphate is a type of element. So, and this, the whole reason behind its regulatory function there is that it helps recruit substrates for mTORC1, so it can bind to substrates. MLST8, this is what binds to the kinase domain of mTOR, and the whole purpose of this is to maintain the interaction between raptor and mTOR, because receptor is what regulates the mTOR activity, and it helps bring the substrate to join onto mTOR to increase or stimulate activity. And Pras40 and deptor, this is what negatively regulates P3ORC1 activity D. So this is why you got like the Holtz sign either side. Now the mTORC2, this is mammalian target of rapamycin complex in 2 there, and it's also otherwise known as the mechanistic target of rapamycin complex 2. And it, as you can see, it has similar structures to mTOR1. For example, it has the mTOR protein. It also has the MLST8, okay, and it also can be hindered by deptor. However, it has additional roles here, or structures. For example, it has receptor rather than receptor, and the receptor is a protein that is associated with the mTOR, and the MLST8, it helps form the complex between the two mTORs, and the MSINR, this is what helps facilitate mTOR activity to phosphorylate its target, AKT. And it's important to state that where phosphorylation takes place, because protein has a long chain of amino acids. It's a polypeptide. So the position where it gets phosphorylated for AKT is at serine residues SER473. So it phosphorylates AKT at that position, and also other kinases. Now the ProTOR1, which is an abbreviation for the protein observed with receptor1, this is what interacts with the receptor, okay. And the HSP70, this is a heat shock protein, and under standard conditions and after the treatment with heat shock, it's necessary to form the mTORC2 and help maintain the activity. Deptor is what negatively regulates mTORC1 as well as mTORC2. So now let's discuss the function of mTORC1 and mTORC2. So the mTORC1 has a regulatory activity, so it regulates different types of proteins involved in various cellular processes. So for example, growth factors, also metabolism, for example with enzymes such as ornithine decarboxylase and glycogen synthase. It also helps to regulate cell growth and proliferation, and this can be achieved via STAT3, so signal transducer and activator of transcription 3, and protein kinase C delta PP2A, so protein phosphatase 2A. So these are examples of downstream signaling involved within cellular growth. Now, growth can be inhibited by cyclin-dependent kinase inhibitors, and two key examples is P21 CYP1, P27 KIP1. Retinoblastoma is a type of tumor suppressor protein, and it helps to normally it helps to regulate cell cycle progression and differentiation. Cellular stress, so for example oxidative stress, this is where you have HI3-1 alpha that has a significant impact on different cellular processes to help maintain the the status. The protein synthesis, this is involved, for example, the ribosomes is where proteins are being made, and protein synthesis is negatively regulated by EGFR, which stands for eukaryotic elongation factor 2 kinase, which has a role within elongation. Cell death. So you have the autophagy, which is like a recycling system, and there's also apoptosis, which is cell death. And this can be facilitated by different members of the protein kinase family, such as protein kinase C delta and protein kinase C epsilon. There's also a lipid synthesis, for example, lipin. So lipin is similar to AKT. It has 3 members within its family, and what it does, it helps regulate the transcription of genes that are involved in lipid biosynthesis, in other words, lipid production. Production of nucleotides. So DNA is a polynucleotide, RNA is a type of polynucleotide, and the structure of nucleotides consists of the phosphate group, the sugar, the ribose sugar, and also the nitrogenous base. Cell migration, that can be facilitated by cytoplasmic linker protein 170. Regulatory function of mTOR-C2. There is similar overlaps. For example, there's growth factors involved, cell death, apoptosis, protein kinase C alpha is involved, and the PKC alpha is also involved in the inflammation process. And there is GTpases, so GTpases of RAC and RO. This helps in terms of cell survival, migration, and regulatory function of the cytoskeleton, particularly of proteins such as actin. Protein kinase C alpha negatively regulates cell cycle progression because in some cells, but in the majority of the cases, proliferation differentiation does take place in most cells. So how is mTOR regulated? Now, there is a positive regulation and there's also a negative regulation. So if it is a positive regulation, AKT is what inhibits TSC complex, and this is what helps increase the activity of PI3K AKT signaling pathway. Now, the negative aspect is when the complex of TSC, which comprises of 2 types, TSC1, TSC2, and a protein called TBC1D7. So what happens is there is an enzyme called a liver kinase B1, LKB1, and it phosphorylates AMPK, which stands for AMP activated protein kinases, and AMPK is what phosphorylates TSC2 protein, and this is what generally negatively regulates mTOR. We'll now progress on to the first step within the cell signaling pathway. So what are the types of examples of first messengers or ligands that are able to bind to the cell surface receptors such as EGFR and RTK, so receptor tyrosine kinases? There are hormones, peptide hormones such as insulin, cytokines, for example, tumor necrosis factor alpha, nutrients, for example, amino acids and glucose, and growth factors via RAS and PI3K. So the first step with any cell-to-cell communication with any signaling pathway, it requires a receptor activation, whether that receptor is found on the cell surface or whether the cell is intracellularly, i.e. inside the cell. So there is a specificity and sensitivity in the interaction between the ligand and the receptor. For example, when you have a growth factor bound with a GPCR, that is what forms the ligand receptor complex. When you have tyrosine kinase bind onto tyrosine kinase receptor, that is what facilitates the ligand receptor complex. So here you can see the plasma membrane, and you can see there is 2 EGFR. So you can have receptors that join together that are homodimers or heterodimers. So in this case over here, we've got 2 EGFR family subtypes here, and there is the ligand binding site at the top. There's also the intermediate location, and then there's the phosphorylated sites at the bottom that have a direction towards inside the cell. The next step is that once the ligand binds with the receptor, dimerization of the receptor occurs. So that means that there's closure or bringing two receptors together. So you can see the phosphorylation sites are autophosphorylated, and you can see the binding between the receptors there. The next step is, after receptor dimerization, the regulatory P85 subunit of PI3K is what binds onto the receptor. So a gentle reminder that the P85 subunit consists of the CSH2, the ISH2, the NSH2, the BH, and the SH3. Then phosphorylation of the PI3K takes place. And there is interaction between the 4 domains during basal states. The basal state is basically the normal state, okay? And the interaction between the domains of P1110 and P85 takes place under normal status. So you could have ABD with CAT, you can have ABD joined with ISH2, you can have the helical join onto NSH2, and you have the RBD that can join onto the ATP site of the kinase domain. And that the last one is what inhibits the activity on the basis of this interaction. Let us now proceed onto the signal transduction. So far, we've got an activated receptor, dimerization took place, phosphorylation of the PI3K takes place. Now, the P10 subunit, which is a catalytic domain of PI3K, produces PIP3 from PIP2. Now, PIP3 is an abbreviation for of phosphodiol-inositol triphosphate. This is otherwise known as the PTDLNS 345P3. Now, the PI3K, in order for this to take place, it phosphorylates the 3-OH group that is present within PIP2 in order to make PIP3. So, phosphorylation does take place. So if we look at this image here by Gribbon and Joel, you can see in the PIP2 where the OH is in the position of 3, via the PI3K, where phosphorylation takes place. In the PIP3, you can see that there's a phosphate group that's attached under the position 3. Now the PI3K can also indirectly stimulate the production of PIP2. Step 6. So the serine threonine kinase AKT is translocated to the plasma membrane, and whilst it's there, it helps phosphorylate or become phosphorylated by 2 enzymes to initiate AKT activity. In other words, PDK1 and mTORC2 are the 2 enzymes that help AKT activity. So let's start off with PDK1. So at threonine 308 position on the kinase domain, and it becomes activated. The active PDK1 can also autophosphorylate at position S241 per position. So that's the serine 241. Now, mTORC helps with the regulatory process. So that's serine 473 in the tail domain. That is what helps AKT to become activated. Now there are other PI3K targets. Okay, so all of them have a pH domain. So RHO, RAS, RAC, ARF, GAB1, 2. Now how does the AKT become negatively regulated? Well, you have 2 structures here, P10 and PHLPP. P10 is an abbreviation of phosphatase and tensine homolog, and the purpose of this is that it converts PIP3 to PIP2, and this prevents the AKT from moving to the cell membrane for further signaling. The other negative regulator of AKT is pH domain leucine-rich repeat protein phosphatase. So what PP2A, it removes the phosphate or dephosphorylates AKT, and this occurs at position of serine 473 and T308. Now the downstream targets of AKT consist of FOXO, which is a transcription factor, GSK3, TSC2, and PRAS40, and there is also P27, which is a type of cyclin-dependent kinase inhibitor. Let us begin with TSC2. So TSC2, when it undergoes phosphorylation, that is what prevents the complex between the 2 types of TS. And the GTP-PASE Reb is what becomes bound with the GTP, which is an abbreviation for guanosine triphosphate. Now, as a result of phosphorylation of TSC2, mTORC1 is what gets activated, and this occurs at position of serine 2448. mTOR inhibits the protein 4E-BP, and the normal role for 4E-BP is to inhibit translation of the proteins. Its inhibition is what promotes the translation of cyclin D1 and helps with the G1 phases, because the cyclin D1 specifically joins with cyclin-dependent kinase 4 and 6. The enzyme CDK4/6 can also phosphorylate the tumor suppressor protein called retinoblastoma, And this is what releases the E2F transcription factor. Now, E2F it can be found within the transcription of cyclin E that will help within the G1S phase with CDK2. PRAS40. So PRAS40 negatively regulates mTOR-C1. Now, when this occurs, AKT is able to phosphorylate the inhibition of PRAS40, and it's further facilitated by red protein. GSK3 beta. This is an enzyme that negatively regulates cyclin D1, and it also degrades the cyclin D1. FOXO. So FOXO is a transcriptional activator that helps to regulate effectively the cyclin D1. And the FOXO has an ability to stop the cyclin D1 gene transcription. And FOXO3 can assist with apoptosis, and this is by increasing or upregulating the genes that are for apoptosis. So FOXO3 helps increase the expression of death receptors. So for example, you have Fas ligand and tumor necrosis factor-related apoptosis inducing ligand. Amongst the downstream targets of AKT is also P27-KIP1. This further inhibits or prevents cyclin D when it bound with cyclin DK4/C. And it can be also phosphorylated by c-MYC, ERK, MAPK. So essentially it's like the protein kinase cascade taking place here. Now, other CDK inhibitors are P21, CYP1, RAF1. So what happens next? So now you have a phosphorylated AKT, and the mTORC signaling pathway is taking place as we speak. There is interaction between receptor of mTORC1 and the TOR signaling motif TOS found in S6K and 4EBP. And the TOS motif comprises of 5 amino acids. So on the N-terminal side of SCK1, you have the 5 amino acids, so 3 ASP, ILE, ASP, LEU. Now on the C-terminus of the other side of 4EBP1, you have 3 GLU, METS, ASP, and ILM. The targets of mTORC1 on the basis of the regulatory function that takes place. The 3 main examples are SCK1, 4EBP1, and P70 SCK1. So SCK1 is phosphorylated at position of 389. And once it's phosphorylated, it is able to phosphorylate another type of protein called S6. So that consists of 4S ribosomal protein S6. So this is what further increases the translation of mRNA sequences for specific types of proteins, and it will comprise of elements in the 5 untranslated terminal regions in target genes, for example, growth factors, protein hormones, and ribosomal proteins. Now, besides mTORC1 and S6K1, it can be phosphorylated by PDK1 and MAPK. Now if you look at the second target is 4EBP1. mTOR negatively regulates translation, and this is achieved by deactivating EIF4E. So that will stand for eukaryotic translation initiation factor. And E1F4E is dissociates or keeps separate from the 4EBP1. And the unphosphorylated of 4EBP1 helps to bind to EIF4E to help prevent translation. Now the P70S6K1, the role of this is mTOR phosphorylates this type of kinase. Amongst the STAT family, you have STAT1 and STAT3. So these are also targets of mTORC1, and it will also help activate the nuclear receptor PI3K gamma. So let's look at the cellular response. So there's a range of different processes that takes place. For example, cell proliferation, cell survival, translation of the mRNA sequences to encode the actual protein, the production of ribosomes, migration, cell growth and size via S6K1 and 4EBP1, cellular metabolism, so for example glucose, amino acids, lipid metabolism, and how to maintain levels within the actual human body. So for example, maintaining insulin levels. And gene transcription, for example, STAT1 and STAT3. So autophagy, so when mTOR phosphorylates EGFR, this is what leads to mTOR being prevented, and this is in order to activate EGFR and induce autophagy. Here we have an image from Creative Commons 2023, and it basically summarizes or has a signaling pathway. So what we can see is a growth factor activates the tyrosine kinase receptor. You can see dimerization of the receptor taking place. The PI3K is involved in converting PIP2 to PIP3. This then leads on to PDK1. Then you have AKT. So AKT, this is what holds the TSC complex. And then the ERK1 and 2 can also inhibit the TSC1-2 complex. After this, we got the mTOR, and mTOR can stimulate the genes that are involved in different cellular processes. So you've got ROA, PKC alpha, Paxillin, SGK, NDRG1. So let's now look at the link between EGFR and PI3K. Now, even though the start is the same, for example, you can have EGFR join onto another EGFR, or you can have EGFR with her family, HER1, HER2, HER3. The downstream signaling is what changes. So with the EGFR, you have RAS and you have RAF1. But with the PI3, AKT, mTOR, it's different targets there. But did you actually know that the PI3K is able to interact with the RAS. So let's discuss. The binding of PI3K with ERBB3 and ERBB4 is via the P85SH2 domain. So you can directly bind. There's also indirect binding of PI3K. So this is also by EGFR family, so more specific ERBB1, ERRB2. And it's facilitated by the adapter protein GAB1. So GAB1, the protein-rich domain that is apparent, binds with the GRB2SH3 domain. Now the GAB1 is phosphorylated at several sites where P85 subunits of PI3K binds. Now the oncogene RAS can also bind to PI3K, and this is via the P110 subunit. The CBL can bind to the P85 and PI3K, and this is necessary to help in terms of EGFR. So let's now look at the dysregulated version of the story of the PI3K AKT mTOR. Now, similar cases of other previous signaling pathways where there is an increased gene copy number or amplification, or if there is a type of missense mutation that took place, this will essentially can lead to a dysregulated pathway and influence the cell-to-cell communication. But let's have an insight into this. So when you have a missense mutation within the PH domain of AKT1, this more or less can be affected. For example, the E17 mutation increases the link of AKT and PIP3. The loss of the tumor suppresses the P10. Overactivation of receptor tyrosine kinase, similar case with EGFR. There is also missense mutations, for example, deletions of expression of EGFR in various cancers. There is also amplification of AKT1 was discovered to be 20 times. E54K and E54 mutations, that's also been found on the P110 subunit of PI3K, and this substantial can help increase activity. Now when you inhibit AKT or mTOR, what happens is the increased activity becomes out of balance, and as a result it can lead to continuous signaling of PI3K and RAS. So let's now discuss the AKT family of when it becomes mutated and its association with cancer. And the presence of the AKT mutations is associated with poor prognosis and more of a advanced stage of the disease. For example, mutations within AKT1. There is evidence that high levels of this specific mutation is found within the bladder, ovarian, gastric and prostate cancer, and 80% of high-grade carcinoma cases. AKT mutations. So when there is undifferentiated cells being apparent, especially within the prostate, ovarian and breast cancer, it accounts for 30 to 40%. AKT3 mutations, This is mainly found within estrogen receptor negative breast cancer. So you have a variety of different genes that are involved in how breast cancer can occur. For example, you've got BRCA1, BRCA2, you've also got the HER family, and you've also got the estrogen receptor. And besides being high levels there, there's also androgen insensitive prostate cancer. Now, the phosphorylated AKT has many functions in terms of cancer hallmarks. For example, its ability to proliferate in a tumor state, invade more tumor cells and locations, metastasis, angiogenesis, evading apoptosis, reprogramming of the metabolism. Let us now look at AKT and evading apoptosis. So for method 1, when there is AKT has ability to phosphorylate FOXL transcription factor, this is what decreases the expression of apoptotic genes. And overexpression of AKT inhibits FOXO3 expression. And method 2 that links both of these factors together is AKT has the ability to phosphorylate BAD. Now BAD is an abbreviation of BCL2 associated agonist of cell death. And this occurs at position serine 136 or 112. The permeability of mitochondria can also be changed, and this helps prevent the cytochrome C release from the mitochondria, which prevents apoptosis. Now, if we reflect back within the previous episode, we discussed that apoptosis has two methods, one that is intrinsic and one that is extrinsic. The third method that links AKT and evading apoptosis is to prevent caspase activity. So AKT can phosphorylate XIAP at position serine 87. AKT has also an impact on invasion and metastasis, both in a normal and mutated way. Now, in terms of the mutated sense, the matrix metalloproteinases, they are a type of that helps degrade the extracellular matrix during the invasion stage, and MMP is regulated by AKT. Now, for EBP1, this functions within the translation of transcription factor proteins such as snail, slug, and twist. So this is what upregulates the EMT. Now P70S6K, this essentially promotes the expression of particular types of MMP such as MMP9, and this is what mediates the breakdown of in other words, the proteolytic activity that is found within the ovarian cancer. And AKT expression, when knocked down, affects mTOR. So, by experimental studies, when you knock down the gene of, in this case, AKT, it affects how mTOR actually works. And this is what downregulates mRNA of MMP2 and MMP9. AKT also has a role in other hallmarks of cancer, such as angiogenesis and reprogramming of metabolism. Now, angiogenesis is the ability to provide nutrients via these blood vessels, and that is what helps the cancer to progress and grow. Now, VEGFR, the vascular endothelial growth factor, helps increase angiogenesis and tumor survival because it activates the PI3K AKT pathway, more specifically the VFL K1 VEGFR2 PI3K AKT. Glucose metabolism is also important because it helps in terms of respiration and the production of various macromolecules. Now, AKT upregulates glucose transporters because glucose can't travel alone in the blood. So what happens is it needs to have a transport protein to facilitate this case. And this is what increases the glucose uptake within cells. And not only this, but there's also an uptake of lipids, glutamic acid or glutamine, and nucleotides. And a combination of all of these is what helps the tumor to grow, what helps it to proliferate, what helps it to differentiate. mTOR also has a role with cancer. For example, inhibitors of mTOR, it increases expression of growth factors, and this influences the activity of PI3K and RAS. mTORC is sensitive to rapamycin, and as a result, it helps to activate S6K1 and 4EBP1 to continue with translation of the proteins. mTORC2 is resistant to rapamycin on the other hand. What it does, it helps stimulate the PKC alpha and AKT to regulate actin cytoskeleton. The effector proteins of mTOR, such as EIF4E, S6K1, and 4EBP1, and overexpression of this is what causes transformation of the cells. PI3 also has a link with cancer, so via the P10 alpha catalytic subunit, it is the most common site where most mutation takes place and found with many types of cancers, such as gastric or the stomach, the endometrial, so the lining of the womb, the ovarian, the brain, and blood cancers. And this is what helps tumor progression, angiogenesis, and metastasis. The regulatory subunit genes PI3K1 and PI3K2, mutations within these areas also increases PI3K activity. P10 and cancer also has an impact within the signaling pathway. For example, the P10 normally negatively regulates, and it's a tumor suppressor, and when there is a mutation or downregulation of P10, this has been evidently shown within prostate cancer, melanoma, which is a type of skin cancer, and non-small cell lung cancers. So by the end of this lecture, what you should understand is that following activation of EGFR or TK1, PI3K is activated. PI3K consists of two subunits, P10 alpha catalytic subunit and PA5 regulatory subunit. The P110 subunit produces the second messenger, PIP3. AKT is a serine threonine kinase that is activated by PTK1 and mTORC2. mTORC1 is activated by phosphorylated AKT. RAS can bind to PI3K, and this is by the subunit P110. Inhibitors of AKT or mTOR increases the expression of growth factor receptors, and this essentially increases activity of both PI3K and RAS signaling. and this is what is associated with poor prognosis. MMPs, they are regulated by AKT, and a knockdown of AKT influences mTOR, and this has an impact on the expression of MMP2 and MMP9 and the overall process of invasion and metastasis. Here is a reference list for further reading. Next week we will discuss PRC gamma 1 and PKC and its association with other signaling pathways. I hope you enjoyed this episode and enjoy the rest of your week. Take care. Bye for now.